So I've demonstrated both common and uncommon evolutionary changes on the nanoscale during a single infection in a single patient. What about the microevolutionary changes? Here I'm talking about the variation in HIV since it first jumped to humans sometime at the beginning of the previous century, between 1900 and 1950. I should start by pointing out that when we say HIV, we usually mean HIV type 1. There's also HIV type 2, which likely evolved independently in a completely different jump from a different animal source. HIV-1 appears to have jumped from chimpanzees to humans, and HIV-2 from sooty mangabes. HIV-2, for the most part, is confined to Africa. It has a very similar, but not identical, gene structure and life cycle. Among HIV-1, there is a division called groups. Group M, which stands for major, represents 90% of all HIV infections globally. Among Group M, we subdivide further into subtypes. If you are in Southern Africa, China, or India, the dominant subtype is designated subtype C. In North America, almost all cases are going to be subtype B. Subtype J is emerging as dominant in the Caribbean, and D in Russia. Each of these different subtypes varies ever so slightly from other subtypes. Given enough time, these differences are increasing. Recent reports suggest that subtype D in the infected population of Uganda has an accelerated disease progression. It is a more virulent virus that is also spreading more quickly and causing worse disease. Subtype B, accounting for almost all disease in North America, shows higher efficiency transmission by homosexual sex than the other diverse forms, which may explain the initial epidemiology of the disease in the US. This is how evolution can tell us the likely future course of the global HIV epidemic. By understanding that HIV is evolving in different parts of the world, we'll be able to adjust the sensitivity levels of diagnostics to prevent the most disease and save the most lives. There's another set of changes we should address on the microevolutionary phase: drug resistance. This has already been discussed so many places, but our current therapeutic strategies are based on an understanding of how natural selection acts on an evolving population. In the case of HIV, we initially only had a single drug that acted on the ability of the virus to copy itself. Why that drug, and not something that acted on the entry of virus into cells? Because we had already determined that the entry process was highly mutable and evolved very quickly. The viral replication mechanism, reverse transcriptase, was something we could determine was highly constrained by evolution. What made it work was a specific shape of the enzyme. Any alterations made that enzyme inactive. On the other hand, the surface envelope molecules we talked about earlier continue to work at least weakly with major alterations. It's much harder for the virus to evolve a resistance to drugs targeting replication than it does for them to evolve new mechanisms of entry. We also mapped the variations in the viral proteases, which were also highly conserved throughout evolution. This led to the development of protease inhibitors, drugs which targeted these highly conserved enzymes. Any one of these single drugs could still be bypassed by evolutionary change very quickly. HIV has a remarkable ability to innovate new pathways, which makes sense because it undergoes both extreme diversity and extreme selection. By combining 3 drugs or even 4 into a single cocktail, we set a higher hurdle for evolution. It must now innovate 4 separate times to be successful. Sadly, even this innovative strategy doesn't work forever. Eventually, a virus emerges which is resistant to all of the drugs, and the patient gets no further relief. Now we're back to the funeral scenario. In a study of female bar and hotel workers in northern Tanzania, 7% of HIV samples showed a primary resistance to the AZT class of drugs, the RT inhibitors, and 7% had resistance to all the protease inhibitors. Statistically, we would then expect about 0.5% to be completely resistant to the triple cocktail. Lastly, let's look at the macro scale of evolution of lentiviruses. HIV is in this viral family, formerly called the Lentaviridae, which includes these closely related viruses. FIV, or feline immunodeficiency virus, which causes immunocompromise in cats, which is found in almost 90% of healthy wild lions in Africa, but is almost invariably fatal in domestic cats. There's also the bovine immunodeficiency virus found in cows, the puma immunodeficiency virus found in New World cats, the more distantly related CAEV found in goats, the equine infectious anemia virus found in horses, and the Visna virus found in sheep. All of them cause some form of immunopathology or disease through disorders of the immune system. All of them have the same basic gene structure and life cycle. We've already mentioned the likely origin of HIV from the simian immunodeficiency virus, another important lentivirus, possibly having jumped from other primates or monkeys to humans multiple times. The level of macroevolution in these viruses tells us how they are related. It gives scientists a way to find the origin of HIV. This was my work, reconstructing the phylogeny of lentiviruses. It's one of the big questions in all of virology. Where do viruses come from? In the case of HIV-1, we have 3 samples to compare. One is taken from a wild chimp colony in Western Africa. The other is an old clinical sample taken from a 28-year-old woman living in Kinshasa in 1960, and the very oldest HIV sample to date collected in 1959 in the vicinity of Kinshasa. The 3 are not quite identical. Of the portion they were able to sequence, the difference between the 2 human samples collected 1 year apart is about 12%, and differed from the chimp sample by about 30% in that same region. From these comparisons, and knowing what we do today about viral evolution, that puts a rather precise date of origin for HIV-1 at 1908, with a 95% probability that the origin was between 1884 and 1924. So evolution as a tool, as a model, and a framework means that we understand this pathogen better. We know where it came from, chimpanzee SIV. We know when it jumped to humans. More than that, though, a comparison of the genomes of all the lentiviruses has led us to a major breakthrough. One of the key differences between SIV and HIV-1 is the type of accessory gene products, and particularly one called VPR, or viral protein R. The same sequence exists in SIV and FIV and EIAV, but it's far more mutable there. We find more variation in it. In HIV, it's highly conserved. That led scientists to investigate it more closely. To early examination, it was a minor, unimportant viral protein that was apparently too small to do much. But the lack of sequence diversity and the alignment to viral genes in SIV and HIV-2 led them to discover the truth about this protein. It has at least 4 distinctive functions that make HIV so deadly. It does things like stopping the growth of immune cells, transporting the virus into the nucleus of the cell, and it regulates viral expression. The ancestor of HIV lacks some of these functions, so it's likely the product of recent evolution. It's an amazing target for gene therapies, and it's an understanding of how the lentiviruses have evolved in response to their hosts that led us to identify that VPR even mattered or played a role in the cell. We've taken a look at 3 different levels at which evolution operates on HIV. What I hope I can call your attention to in each of these levels is that evolution is not being asserted. Evolution is the model, the tool that scientists and medical researchers have used to make major strides in treating a deadly disease that tears apart family and kills the young and innocent. You asked for proof that evolution is a fact, as though that were something that modern biologists were really apprehensive about. The proof of evolution is in the fact that it is a predictive model of practical utility. It's like the theories of physics to an engineer. The idea of contesting something that can be used to hit an asteroid millions of miles away with a small probe launched from a rocket never enters their mind. They work with the theories of physics every day. Likewise, evolution has been described as the unifying theory of biology. Nothing in biology makes sense except in the light of evolution, as Dobzhansky so famously said. That's the reality. Now, I have little or no hope of changing the mind of even a single creationist, yourself included. I don't think this is something you are prepared to be logical or rational about. You've internalized your creationism, and it would be too painful for you to question this core belief. But this gave me an opportunity to tell you why I oppose your attempts to inject religious dogma into science education. It's because every day, molecular biologists show up to their labs. They work for little pay on problems that reduce suffering and pain, make life on Earth a little brighter. They use evolution to do that. I know you think you're doing a good thing, trying to get your religious beliefs into that lab by way of the educational system or public funding choices. But I'm here to tell you that you're only going to replace a workable model of genetic change with something that doesn't answer any questions, except how much you're willing to let others suffer for your religious beliefs. I think that's an ugly and an evil thing. Thanks for watching.
Every cell of each plant and animal contains genetic information coded onto the DNA molecules.